In this video, I'm going to talk about animal embryo development. It's also called embryogenesis. And the reason it matters to biologists and to students is because it allows us to very readily determine commonalities and differences between animals in the animal kingdom, for instance. If we were to look at a chimp embryo and a human embryo, what we would find is that they are extraordinarily similar to each other. And that, in addition to DNA evidence, provides us a lot of evidence to suggest that humans and chimps are very closely related. And looking at an embryo or studying the embryo development is a lot easier to do than sequencing genomes, for now at least. And so that's the reason why embryogenesis has been so important to biologists in the past, and why it's important to know, especially if you're learning about biology, because it helps us a lot, especially when we're making cladograms. And so with that, let's get into it. And the first thing we're going to note is after the sperm fertilizes the egg, we're going to end up with a zygote. And zygotes are diploid, 2N. That means that they have 2 copies of every chromosome. And that comes half from the sperm, which is haploid, as well as half from the egg, which is also haploid. And then after we form a zygote, the next thing that's going to happen is rapid mitotic divisions. And the thing with mitosis is that we get exact copies of the parent cell in the daughter cells. And what we'll see is that at the 8-cell stage, we have 8 identical cells, assuming we have radial cleavage. So this radial cleavage that occurs on your zygote is one of the key features that we find in animal embryos. And this is one of the things that helps biologists identify if it's an animal just by looking at the embryo. So it's pretty cool. Now, the thing with radial cleavage is it's also referred to as indeterminate cleavage. And the thing about indeterminate means that all 8 of these cells are identical. And because they're identical, they can go on to become any cell type and exist in any tissue. So at this 8-cell stage, we have stem cells. And there are many ethical dilemmas presented here, but for the sake of introducing a biology concept, this is where stem cells are coming, the 8-cell stage, assuming we have radial cleavage. And then what's going to happen next is we're going to continue our mitotic divisions and form something referred to as a morula. And the morula exists between these 2 steps here. And the morula is just a solid mass of cells. And the reason it's important to consider the morula is because there are species and organisms that stop embryogenesis essentially at the morula. Once they reach the morula, in the case of sponges, they just continue to grow, and that's all you see. But in the case of more developed organisms, we see something occur afterwards, referred to as blastulation. Blastulation. And I like to think of blastulation as the formation of a basketball-like embryo. And what I mean by that is it's now quasi-hollow. It's filled with fluid, but what we have now is this kind of cell lining that's just 1 cell thick. And what we get out of it is a blastula. And the inside of the blastula is called the blastocell. Now, with the blastula, the next stage is going to be a process referred to as gastrulation. And gastrulation I like to think of as infolding. And so in this infolding process, what's going to come out of it is something referred to as a gastrula. Now, the reason we care so much about the gastrula is because we're seeing the formation of things referred to as germ layers. So you're going to see a lot of G's. You get germ layers. And these germ layers will go on to become tissues. And so if we were to take a closer look at a gastrula, what we're going to find is on the outside, we are going to have something referred to as an ectoderm. And that's the outermost cell lining. Now we have an inside cell lining, and this inside cell lining is referred to as an endoderm. And the endoderm will become the digestive lining later on in development, depending on what kind of species you're looking at. In addition to that, we still have the blastocele, the inner cavity that existed within the blastula. But now what we've formed, thanks to the gastrulation process, is something referred to as the archenteron. And the archenteron has this kind of pore opening to it that we refer to as the blastopore. And at this point, what we have is something referred to as a diploblastic embryo. And diploblastic here refers to the fact that we have 2x germ layers, which was our endoderm and our ectoderm. And for an introductory embryogenesis discussion, this is where sometimes people will stop it, because we will see more branching off as we continue. And to allude to some of that, particularly in the case of humans, you might ask yourself, well, where does your body cavity come from? And where's your heart, your liver, kidneys, where's all that stuff going to be stored within this embryo? And so at that point, what's going to happen here is, if we're working with a triploblastic embryo, then a mesoderm will form. And these mesoderms are your body cavity. And in the case of humans, the mesoderm will form by forming these kinds of buds that will eventually merge together later on. But we also see a mesoderm forming by these kind of hollow regions here that will also fuse together later on. So we see these kinds of diverges or these kinds of forks in the road as we're following this embryogenesis path. And that's the reason why it's so useful to us as biologists in creating cladograms and classifying organisms and trying to find out how life is related to each other. And so this is going to wrap up my introduction to embryogenesis. I hope you guys find it useful. Let me know if you have any questions, and thanks for watching.